Welcome to this lecture on immunology. Today, we are going to explore a fascinating and fundamental concept, B-cell and T-cell epitopes. Imagine trying to recognize a friend in a crowded room. You do not need to see their entire body, just a familiar face or a unique feature. The immune system works in exactly the same way. It does not recognize an entire massive bacteria all at once. Instead, it looks for tiny, specific physical spots to attach to and neutralize the threat. Let us dive in and see how this works. Now that we have our analogy, let us define what these recognizable features actually are. An epitope, formerly known as an antigenic determinant, is the specific, localized region on or inside an antigen. When a large pathogen enters the body, only these tiny epitope regions make physical contact with our immune receptors. For proteins, an epitope is usually just a very short sequence of amino acids, or a specific folded shape. Because a single antigen is huge, it can possess many different epitopes, triggering a diverse, multi-targeted immune response from our body. Understanding these tiny regions leads us to a crucial question. Why do epitopes matter so much in medicine? Epitopes form the absolute fundamental basis of immune specificity and immunological memory. Without them, our immune cells would have to engulf and analyze entire pathogens, which is far too slow to keep us alive during an infection. When scientists design life-saving vaccines, they are essentially introducing safe versions of these specific epitopes so our bodies can learn to recognize them. Furthermore, mapping epitopes helps doctors develop targeted therapies for autoimmune diseases, where the body mistakenly attacks its own healthy cells. Having established why epitopes are important, let us look at the first major type, B-cell epitopes. B cells are the immune system's long-range snipers. They produce antibodies that float through the blood to hunt down intact, living pathogens. Because these antibodies encounter pathogens in their natural, fully folded state, B-cell epitopes must be located on the outer surface of the antigen. Imagine touching a closed puzzle box. You only feel the exterior. Consequently, these epitopes are typically hydrophilic, meaning they love water, which makes perfect sense since they are exposed to the watery environment of our blood and lymph. Building on the idea of surface epitopes, we must understand their physical shapes. B-cell epitopes generally come in two forms. The first is linear, which consists of a simple, straight line of sequential amino acids. The second, and very common type, is conformational. In a conformational epitope, amino acids from completely different parts of the protein chain are brought close together by the complex three-dimensional folding of the molecule. If the protein unfolds or denatures, a conformational epitope is destroyed because those specific amino acids are no longer held next to each other. Now that we know what B-cell epitopes look like, let us examine how well an antibody actually recognizes and holds on to one. We measure this chemical attraction using the association constant, denoted by K. Conceptually, this formula tells us the ratio of successfully bound pairs to the remaining unbound pieces floating around. C sub B is the concentration of the bound antigen-antibody complexes. C sub F is the free antigen, and C sub A is the free antibody. If the value of K increases, it means the antibody binds very tightly, which is ideal for a strong immune response. Let us apply this formula with a concrete example. Suppose a researcher is testing a synthetic B-cell epitope for a new vaccine. We are given the free epitope concentration as 0.002 molar and the free antibody as 0.005 molar. The bound complex is 0.05 molar. We plug these values into our equation, dividing the bound concentration by the product of the free concentrations. The denominator becomes extremely small, 0.00001. Dividing 0.05 by this gives us a final association constant of 5,000 liters per mole. This high number physically means the antibody binds very securely, making it an excellent vaccine candidate. We've seen how B cells attack the outside of pathogens, but what about the inside? This brings us to T cells, which act as the immune system's strict internal inspectors. Unlike B cells, T cells absolutely cannot recognize whole, intact antigens floating in the blood. Instead, the antigen must first be consumed by an antigen-presenting cell. This cell acts like a chef, taking a whole cake, chopping it up into tiny slices, and serving it on a plate. Because the protein is completely dismantled, T cells get to inspect the hidden internal components of a virus or bacteria that antibodies could never reach. Following up on how these fragments are prepared, let us look at how they are displayed. The antigen-presenting cell uses a special molecular tray called an MHC molecule to hold the chopped-up fragment on its surface. Because the original protein has been completely unfolded and sliced, T-cell epitopes are always strictly linear sequences of amino acids. They are never three-dimensional conformational shapes. Furthermore, because the protein is dismantled, these epitopes can come from anywhere, including the deep, water-hating, hydrophobic interior of the pathogen, providing a highly thorough scan of the invader. Since T-cell epitopes are just short, linear fragments, we can mathematically calculate how many potential epitopes a single protein might yield. MHC molecules have strict size limits, requiring peptides of a specific length. This formula finds the number of possible fragments by sliding a window of length L across the entire protein sequence of length N, one amino acid at a time. If the total length N increases, the number of potential targets for the immune system increases. If the required peptide length L increases, the number of possible fragments slightly decreases because a larger window fits fewer times into the sequence. Let us see this sliding window concept in action. A virologist is studying a viral spike protein that is exactly 120 amino acids long. The human MHC class I molecule requires peptides that are exactly 9 amino acids in length. We want to find the maximum number of unique overlapping peptides. We substitute N with 120 and L with 9. Subtracting 9 from 120 gives us 111, which represents the sliding room. Adding 1 back for the starting position gives us a final answer of 112. This means the immune system has 112 different possible fragments to scan from this single small protein. Now that we have calculated both types, let us look at a visual summary of how they differ. On the left side of this diagram, you can see the B cell mechanism. Notice the large, intact, folded protein. The Y-shaped antibodies are binding directly to the outer surface, recognizing both linear and complex three-dimensional conformational shapes. On the right side, we see the T-cell mechanism. The original antigen has been chopped into tiny linear fragments by the antigen-presenting cell. You can clearly see one of these small red fragments being held up by the MHC molecule tray, ready to be inspected by the T cell. To wrap up our journey, let us review the most important ideas we have covered today. First, epitopes are the specific, tiny regions of an antigen that the immune system actually sees and binds to. Second, B cell epitopes are found on the intact surface of pathogens, allowing antibodies to attack invaders directly in the blood, using both linear and three-dimensional shapes. Finally, T cell epitopes are strictly linear, chopped up fragments presented on MHC molecules, allowing T cells to inspect the internal contents of an infection. Together, these two distinct systems ensure our bodies can detect and destroy threats wherever they hide. You now have a solid understanding of immunological recognition.